for reminding me.
You're welcome.
You see the blinking red dot?
I do now.
You should. Okay. Okay, so what was the question? Someone was starting to ask me a question.
Yeah, I was just reminding you about the thing. Was Stuart, I think, was asking something, or I think she was just saying hi.
Okay, well, I missed it, whatever it was. All right. First thing I'd like to do today is share my screen with you and go over the worksheet that you just turned in. Do you see it?
Yes.
Okay. All right, first let me point out that in the assignment, it said that this worksheet was worth 20 points. It's really worth 19. So if you think your score was 1 point too low, that's why. So the first question was 3 processes by which a cell can generate ATP. Most everyone got this right. Photophosphorylation, substrate level, and oxidative. And we're going to talk a lot more today about the latter two, substrate level phosphorylation and oxidative phosphorylation. Number 2, another way of, another source of energy for Earth's life forms is chemosynthesis. And then number 3, there are 2 possible answers here. About half of you had what I was expecting, that photosynthesis consumes energy and is anabolic, whereas respiration releases energy and is catabolic. The other half had endothermic and exothermic. I gave credit for either because either of them is correct. I prefer anabolic and catabolic because endo and exothermic imply generation or consumption of heat, and that's not necessarily involved, at least not primarily involved, in these processes. At any rate, on your sheet, when I graded, if you had said endergonic and exogonic, I wrote in anabolic and catabolic, but you still got full credit. Right, in oxidation reduction reactions, what's true? Energy or electrons are transferred back and forth between molecules called carriers. That's true. So it's not asymmetrical. Substance that gains an electron is reduced. That's correct. The second 2 are not correct. And then the last 3 are all correct. Substance that loses an electron is oxidized. These things occur in pairs. And the whole point is to make ATP. Then number 5, Scott likes to point out that oil rig is a convenient mnemonic for oxidation and reduction. So I ask if that is true, and it is. Then number 6, you had to match, do a matching exercise. Absorption of light was number 5, which is the thylakoid. The Kelvin cycle is 6, chloroplast stroma. Photosystem 1 is also 5, thylakoid. Krebs cycle is 2, mitochondrial matrix. Glycolysis is 3 in the cytoplasm. And oxidative phosphorylation is 7, the inner mitochondrial membrane. Number 7, chemiosmosis is a critical step in ATP production. Which of the following are true? Well, the first one is not true because it does occur in photophosphorylation. Generates ATP by passing ions across a semi-permeable membrane. It's not a catabolic process. It is the source of electrons for chemiosmosis. We'll talk a lot more about that later. It's part of the Kelvin cycle. No, it is not. And an ion gradient is essential for it, and that is true. Almost no one got all of number 8 correct. A couple people did, but most of you missed 1 or 2 of these. So which is indicative of the Kelvin cycle and which the Krebs cycle? So A requires oxygen, that's Krebs. Creates glucose, that's Kelvin. Occurs only in plants, is Kelvin. Produces ATP and carbon dioxide, that's Kelvin. Anabolic, Krebs. Number D is Krebs. E, an anabolic process, is Kelvin. Occurs in a mitochondrial matrix, is Krebs. It's part of photosynthesis, Kelvin. Oxidizes glucose, Krebs. Includes the chloroplast dark reaction, is Kelvin. Part of aerobic respiration is Krebs. Occurs inside in the chloroplast stroma, Kelvin. Consumes ATP and carbon dioxide. A lot of people missed that one. That's the Kelvin cycle. So where consumes ATP and carbon dioxide, where do those two components come from? Where does the ATP come from? Kelvin cycle occurs, yeah, go ahead.
Does the ATP come from the Kelvin cycle?
It's used by the Kelvin cycle.
Oh wait, sorry. Is it from the photosynthesis that occurs before it?
Yeah. And where does the carbon dioxide come from? Remember, this is called carbon fixation. It's part of the dark reaction. So one of the points that I emphasized here is that this is an amazing process that converts a gas to a solid. Carbon dioxide fixation. So the carbon dioxide comes from the air. And finally, catabolic process is Krebs. Okay, the next one, you see a yellow tree. And the question is, how can you explain that this tree is not dying? It's actually growing. And what I was looking for here is that this is really a green tree that has its chlorophyll is masked by a yellow pigment. So if that is the case, how could you tell whether or not this yellow plant is a parasitic plant?
Because it's growing. Sure.
Yeah, parasitic plants grow too. Just imagine yourself looking at this. This is a tree. Now you're looking at this tree, and It doesn't say anything about anything unusual about this tree except it's yellow.
So we can say it's parasitic plant, which feed on a green plant. Yeah.
Well, I'm saying, why do you think it's probably not a parasitic plant?
Because it's growing.
Yeah, well, parasitic plants grow too.
But the green pigment is masked by yellow color.
Yeah, that's the most likely explanation, that it produces a yellow pigment as well as chlorophyll.
Yeah.
Well, I think if you imagine standing there looking at this tree, you see that it has roots. And a parasitic plant would almost certainly not have roots, because it gains all of its nutrition, including water, from its host.
Because of this, it's called a parasitic.
Yeah, if it's gaining all of its nutrients, including water, from its host, it makes it parasitic.
So it is look like bacteria, or we can say virus. That's it's take all the nutrition and DNA from the other cells, from the real cells.
From the host cell.
Yeah.
So there are really two organisms. In the case of a parasitic plant, there are two organisms, one growing entirely at the expense of the other.
And depends on the, yeah.
The parasite will eventually kill the host. It may not do
Yeah.
it quickly, but it just keeps robbing it of nutrients, and the host can't keep up. As the parasitic plant keeps getting bigger and bigger, its demands keep growing.
Yeah, so it's the same scenario, but in different way. I mean, for bacteria and parasitic plant.
Yeah, you're making an analogy between a parasitic bacterium and a parasitic plant.
Yeah.
Yeah, the analogy is good. It is the same. Bacterium gains all of its nutrition from its host.
Yeah.
Okay, let's go on. Energy currency is ATP. Everybody got that. On the next one, I actually made a mistake in wording this. During aerobic respiration, glycolysis yields 4 net ATP. That should be 4 gross ATP molecules. And the Krebs cycle produces 34 for a total of 38. So if I had worded it correctly, it should be true. But because I didn't, I accepted a false answer as correct. Next one's about lions. They don't eat plants, so do they need vegetation? And the answer is yes, because their prey, in all likelihood, eats plants. Somebody eats plants that eventually is involved in the nutrition of the lions. And most of you got something like that in your answer. Respiration and photosynthesis have generally opposite functions, but there are some similarities. What are they? Well, they both depend on enzymes. They don't both fix carbon. Which one fixes carbon? We just talked about it.
Calvin cycle.
Right, which is part of photosynthesis. They occur inside membrane-bound organelles, and most of you did not get number E. They occur in green plants, and that's an important point, because even though we emphasize that green plants are photosynthetic, they make food, they also consume food. They need food just like all other organisms. So they respire. So photosynthesis and respiration occur both occur in green plants. The next one, 14. Feedback inhibition is a term that designates an unusual class of enzymes. Well, down the face of it, that's not true. This was a false statement. Then the next one, acetyl-CoA, is a so-called crossroads molecule. It is the starting point of the Krebs cycle. Can be derived from a wide variety of complex molecules, which include polysaccharides, lipids, and proteins primarily. Now, if you put down nucleic acids, that is not incorrect. It's just that nucleic acids would be a very small portion of the nutrients in the food that we eat. The bulk of what we eat is polysaccharides, lipids, and proteins.
Because teacher, it's say that complex molecules.
Yeah.
Because of this, protein is correct instead of nucleic acid.
Well, nucleic acids are a complex molecule.
Yeah.
So they, if you didn't put nucleic acids, that's fine, because nucleic acids are such a small component of the food that we eat. It's mainly composed of polysaccharides, lipids, and proteins. The next one, chlorophyll, is a plant pigment that captures energy and sunlight by absorbing red wavelengths and blue wavelengths, but not green, which it reflects. And the next, photosynthesis can occur in any part of a plant. That's wrong. It does not occur in any part that is not green, like the roots. Main components of a chloroplast are the thylakoid membrane, not the vacuole, the stroma, not the mitochondrion, grana, not endoplasmic reticulum, the outer membrane, and the thylakoid membrane. So there are 5 elements on this list that are components of a chloroplast. And finally, the chloroplast harvests chemical energy from light through the activities of not 1 light reaction, but up to a maximum of 3 light reactions. That's false. What's wrong with it? You remember the light reactions in photosynthesis? We're going to have to go over photosynthesis again. There are 2, photosystem 1 and photosystem 2. So that's a false statement. Okay, anybody have any questions or comments about this worksheet? Pay attention to it when you study for the quiz on Wednesday. There might be something in this that will show up there. Not sure yet. Okay, if nothing else, I'm going to stop sharing that. And we're back to an overview. You all see it?
Yep.
Okay. Well, we're almost done with this. We've done energy enzymes and photosynthesis. Photosynthesis is what produces food. And now we're down to emphasizing respiration, which is how we derive energy from food. Deriving energy from food can be either aerobic or anaerobic. Anaerobic respiration is called fermentation. So here you see a flow chart of both of them. Anaerobic or aerobic oxidation is called cellular respiration. The first step is glycolysis, which feeds the Krebs cycle, which feeds electron transport, which is where oxidative phosphorylation occurs, where we get the bulk of our energy. Now, what if the cell runs out of oxygen, becomes anaerobic? It's called fermentation. And it can go either of two ways, depending on where it is. If it's in a muscle, it leads to lactic acid formation, and we'll talk about the consequence of that a little later. If yeast or other microbes are involved, it leads to alcohol production plus carbon dioxide. And I'll talk in detail about these two anaerobic processes later. Okay, an overview of respiration. It starts with glucose, undergoes this process of glycolysis, which is a multi-step process to produce pyruvate. This occurs in the cytoplasm. Pyruvate is capable of passing across the mitochondrial membrane and starts the Krebs cycle. And we'll talk about that later. What's important here, two things. One, there's a substrate level ATP production in glycolysis. There's another one in the Krebs cycle, but the bulk of the ATP comes from oxidative phosphorylation via the electron transport chain. So the main function of the Krebs cycle is that it produces reducing power. Electrons carried via these electron carriers provides them for the electron transport chain. We get other electrons from glycolysis, and because there are so many of these, a lot of ATP is produced. So that's the overview. Let's go back and talk about both of these processes of ATP production, substrate level and oxidative phosphorylation. You've seen this before. Substrate level is a direct enzymatic transfer of phosphate from a phosphorylated molecule to ADP to make ATP. And it's a one-to-one ratio. So it can start with a phosphorylated compound, phosphoenolpyruvate. An enzyme called pyruvate kinase takes that phosphate from PEP, hands it off to ADP to make ATP, and pyruvate is the product. Pyruvate kinase, anytime you see an enzyme named a kinase, it almost surely involves a transfer of a phosphate group. Okay, so that's substrate level phosphorylation. This is another look at it. The enzyme is pyruvate kinase. The substrate, PEP, plus ADP. ADP takes the phosphate from PEP, adds it to ADP, and you get ATP. All right, let's take a closer look then at oxidative phosphorylation. ATP from glucose via respiration in the mitochondrion. Electrons from organic compounds like glucose are passed through a series of reactions called the electron transport chain to an inorganic molecule like oxygen. And oxygen is very important here. Chemiosmosis then generates ATP as energy is released. And from oxidative phosphorylation, you get many ATPs. And I show you this little diagram again to remind you what chemiosmosis is. You get a gradient across a membrane, a high concentration of ions on one side, low concentration on the other. Then the passive diffusion of these ions through the membrane in the presence of an enzyme, ATP synthase, generates ATP from ADP. So the definition of chemiosmosis is movement of ions across a semi-permeable membrane down their electrochemical gradient. So let's go through this diagram of oxidative phosphorylation. Again, it starts with glucose, which undergoes glycolysis to yield 2 molecules of pyruvic acid. And when it does that, as we saw just in the previous slide, there's a substrate level production of 2 ATPs, net 2 ATPs, along with 2 NADH and 2 protons. Then conversion of pyruvic acid to acetyl-CoA yields 2 molecules of carbon dioxide, going from a 3-carbon molecule to a 2-carbon. And one of those carbons goes off as carbon dioxide. Again, 2 more NADHs, 2 more protons. And then we have acetyl-CoA, which is the first step in the Krebs cycle. The Krebs cycle then gives more carbon dioxide and more NADH, more protons, plus a different carrier, a flavin, FADH2, to give a large pool of reducing power. NADH, FADH2, plus 10 protons. And in the process of the Krebs cycle doing this, it generates another 2 ATP via substrate level phosphorylation. Now, all of these electrons and protons enter the electron transport chain. And we'll take a closer look at this in a little bit. But this is oxidative phosphorylation yielding a total of 34 ATPs. So we end up then with a total of 38. 34 from oxidative phosphorylation and 4 more from substrate level phosphorylations. Now, the important thing that I want to emphasize here is at the end of the electron transport chain, the final electron acceptor is oxygen. And that is why we breathe. That's why fish flap their gills, because they need oxygen. So an oxygen molecule plus protons yields water. So, and this is in an aerobic environment. So aerobic respiration has this requirement for oxygen. So we're going to talk later about anaerobic respiration, in which case there is no oxygen here. And so if there is no oxygen, what would happen?
Then oxidative phosphorylation would shut down.
Right. The electrons that come down the electron transport chain would have no place to go. So the electron transport chain would be blocked at the end. everything backs up so that the Krebs cycle stops. Everything stops after glycolysis. So that's why the requirement for us breathing air, the requirement for oxygen is so important. I want to dissect the electron transport chain. Again, we've gone over this before, but and you saw a video illustrating this. And if you want to see that video again, it's in the recording from last time that I put in Canvas. But these electron carriers are what composes the electron transport chain. So the basic phenomenon is that you start with a reduced molecule which hands off electrons to an oxidized one, and that one in turn hands off electrons. And a side benefit of that is extra protons. And to make the point here about the electron transport chain, here, where oxygen is the final electron acceptor, if you substitute oxygen for B here, an oxygen molecule is the final electron acceptor, and water is the result. Again, I refer you to the video if you'd like to see this process in action, but it involves the intermitochondrial membrane, which you see here, these protein complexes that are embedded in the membrane, which harbor the electron carriers. So if you start out with NADH from the Krebs cycle, entering the first protein complex, a flavin, and the electrons keep being passed along from one carrier to the next through cytochromes, and finally end up with oxygen. And oxygen then, oxygen plus protons, is converted to water. The consequence of these electron transfers is that protons, every time there is an electron transfer, protons are generated, which go to one side of the membrane, to the intracellular membrane space. So they accumulate to a high concentration, and at some point, they pass back through the membrane into the matrix. And when they do that in the presence of ATP synthase, that's chemiosmosis. That's what generates ATPs. Now, where does all this occur in the cell? This big blue thing is a cell. You can see the various organelles. And the little orange body is a mitochondrion. Within the mitochondrion, there's the membrane system. With the inner system is called cristae, or crista for a single fold. Within that is the matrix, and on the other side of the membrane is the intermembrane space. And chemiosmosis occurs across this inner mitochondrial membrane. That's where the proton gradient is generated. And when the protons pass through this inner membrane, they generate ATP. Let's go back to glycolysis. I've showed you this before, but glycolysis is usually represented as a one-step process. One glucose molecule, which is a 6-carbon molecule, is converted into 2 pyruvates, each of which is a 3-carbon molecule. Well, in fact, it's much more complicated than that. There are 9 steps from glucose to pyruvate, 2 of which consume ATP. So from glucose to glucose 6-phosphate takes an ATP, and another step later takes another ATP. But then as the pathway proceeds, you get 2 substrate level phosphorylations, which each generate 2 ATP. So it costs 2, it produces 4, so the net ATP from glycolysis is 2 ATPs. Well, this is called the grooming of pyruvic acid. And what this means is that you go from a 3-carbon compound to a 2-carbon compound plus carbon dioxide. So glycolysis occurs in the cytoplasm. You it generates pyruvate, which is a 3-carbon compound. This carboxyl on one end becomes carbon dioxide. And then we have these 2 carbons that are in red, which end up over here in acetyl-CoA, a 2-carbon compound with those same 2 red carbons. So the pyruvate can cross the membrane into the mitochondrion. And when it does so, this carboxyl splits off as carbon dioxide, and NADH plus protons are generated. And now we have acetyl-CoA, which is the first step in the Krebs cycle. And that is here, the Krebs cycle. Here is acetyl-CoA, a 2-carbon compound. So the first reaction in the Krebs cycle is that acetyl-CoA, a 2-carbon compound, is combined with oxaloacetate, which is a 4-carbon compound, to produce citrate, a 6-carbon compound. So you can count the carbons, 2 red ones, 2 blue ones, and 2 black ones. Citrate is then transformed to isocitrate. Then the next step releases a carbon dioxide plus NADH and protons to give, because you've lost a carbon dioxide, alpha-ketoglutarate now has 5 carbons. Lost one of the blue ones. This one is lost to carbon dioxide. Alpha-ketoglutarate has 5 carbons. Then it loses another carbon to yield succinyl-CoA, which has 4 carbons. And again, there's an NADH plus protons produced. And when succinyl-CoA is converted to succinate, the CoA splits off and you get another substrate level phosphorylation, or ATP. So this is the only place in the Krebs cycle that ATP is generated. Then it goes on, more reducing power is generated, FADH2. And finally, you're back to oxaloacetate, which can combine with acetyl-CoA to make citrate, and the Krebs cycle starts all over again. So in terms of energy production, The Krebs cycle itself doesn't produce much. Just one step produces an ATP. What it does produce that is much more important for production of quantities of energy are the NADH and FADH2. Okay, is that clear? Everybody following? So let's take a closer look at substrate level and oxidative phosphorylation. Again, if we start up here with the cell, the nucleus and the mitochondrion, take a closer look inside the mitochondrion. We'll talk about all these structures, but outside the mitochondrion, in the cytoplasm, we start with glucose. And where does the glucose come from?
From
Photosynthesis?
Did you say photosynthesis?
Yeah.
Yeah, that's right, photosynthesis. Produces glucose from the Calvin cycle. A dark reaction, remember, it fixes gaseous carbon dioxide into a sugar. So this glucose then is in the cytoplasm. It is converted by this 9-step process that we just talked about to pyruvate. And the net of that process is 2 ATPs from one glucose molecule. Pyruvate then is capable of crossing the mitochondrial membrane. So now it's inside, and it is then converted, as we just saw, where was it? To acetyl-CoA. That's here. And acetyl-CoA is the first step in the Krebs cycle, also called the TCA cycle. And we've just gone over the details of the Krebs cycle, where you get several molecules of carbon dioxide are generated, plus ATP, and a lot of reducing compounds, electron carriers carrying electrons and protons. And it's this reducing power that enters then the electron transport chain, which is in the inner mitochondrial membrane. We've gone over this several times, so I won't belabor it. But what happens is electrons are passed along, protons are pumped from the matrix to the inner membrane space. So you get this high concentration of protons in the inner membrane space. Then they can diffuse back across the membrane in the presence of ATP synthase. And that's what generates ATP from ADP plus PI. And again, oxygen is the final electron acceptor. And I'd just like to remind you that under anaerobic conditions, if there is no oxygen, the whole process stops. So electrons can't flow, and it backs up. The Krebs cycle can't run, and it backs up to pyruvate. And when we get to anaerobic respiration, we start with pyruvate. We will talk in a bit about acetyl-CoA being a crossroads compound. It's a high energy fuel for the Krebs cycle. It acts as a crossroad compound. It's a common pathway point for the extraction of energy from food, from carbohydrates, fats, and proteins. And in that sense, it's catabolic. But it can also be used as a building block for the synthesis of fatty acids. And in that context, it's anabolic. So that's why it's called a crossroads compound. It can be either of these two processes. And this is the structure of acetyl-CoA at the bottom. If phosphorylated ADP, you can add this vitamin, pantothenic acid, and finally acetyl group on the end. That's acetyl-CoA, this high energy compound. So coming back to acetyl-CoA being a crossroads, we've just talked about this catabolic process where it converts, or pyruvate is converted to acetyl-CoA, which starts the Krebs cycle. And that is coming from glucose. It can also, so that's a sugar, it can also come from fat. So triglycerides are fat. They can be converted to hydrolyzed to fatty acids, and fatty acids can be oxidized to acetyl-CoA. So acetyl-CoA can come from two directions. So if you run out of sugar, so there's not enough acetyl-CoA coming from pyruvate, what your body does is start to convert fats to acetyl-CoA. Now, the reverse of this can also happen. So supposing you eat too much sugar, supposing you have a candy bar. So now this pathway through pyruvate is overloaded and you get excess acetyl-CoA, more than the Krebs cycle can accommodate. So this pathway from acetyl-CoA to fatty acids to fat can be reversed. That's what these dotted arrows show. That if there's too much acetyl-CoA, then you make fat. Is that clear?
Okay, teacher, when we get less glucose or less energy, it's caused to triglyceride and fatty acid reduced in our body because of this.
If you're saying if we don't have enough glucose?
If we don't have get enough glucose or reduce the glucose, which make pyruvate and energy, maybe fatty acid reduced in our body because it take part in the Krebs cycle.
Yeah, that's the point here. That if your body is starving for sugar, it probably still has fat, and so it can stop using sugar for acetyl-CoA and starts hydrolyzing fats.
Yeah.
Remember, fats are triglycerides, and fatty acids then can be converted directly to acetyl-CoA. So this pathway can run either direction. If fatty acids are being converted to acetyl-CoA, that's catabolic. You're breaking them down. If acetyl-CoA is converted to fatty acids and in turn to triglycerides, that's anabolic. Acetyl-CoA is a building block for fat.
And most part of the ATP produced by chemiosmosis done in cytoplasm.
Chemiosmosis does not occur in the cytoplasm.
And no, in here, and Krista.
Yeah, right. These little purple figures here indicate chemiosmosis. That's where ATP synthase is located, and those are the channels through which the protons flow to make ATP in the cristae, the membrane, the intermitochondrial membrane. So that's a complete separate process. And what I was trying to do is isolate out the beginning of this process of ATP synthesis and talk about acetyl-CoA having this dual role for the cell, which is either it can be catabolic or it can be anabolic, depending on what your body needs. If your body has too much glucose, then it becomes anabolic. You start making fat from having too much sugar. On the other hand, if you run out of sugar, you can hydrolyze fats to make acetyl-CoA to make ATP.
Am I understanding correctly then that is effectively how weight loss occurs? That you basically are just reversing that, so you're using your triglycerides for an energy source versus consumed calories?
Yeah. Yeah, so if you're too fat, stop eating sugar, and you'll start using triglycerides for energy. Triglycerides being fats.
And then I have one more quick question. So after glycolysis, what enters the mitochondria is not two free electrons, it's the NADH complex, correct?
Right, that's what's shown
Okay.
here. You have, yeah, those
Okay,
electrons.
because for some
can
reason my
cross-
brain was going, oh yeah, it's the NADH and then two free electrons. And I'm like, that doesn't seem quite right. Hang on.
Right, yeah, so the pathway is shown here. Those can cross the membrane along with pyruvate. Okay, catabolic pathways. Stage 1, hydrolysis of complex molecules to their component building blocks, and these are primarily proteins, polysaccharides, and lipids. Proteins can be converted to amino acids, lipids to fatty acids, polysaccharides to monosaccharides. All of these can be converted to acetyl-CoA, and that starts the Krebs cycle. The Krebs cycle then produces ATP, substrate level ATP, produces carbon dioxide, and it produces NADH, which goes to the electron transport chain, which feeds electrons or feeds protons to chemiosmosis. And that makes ATP. So that's going all the way from food to energy. And this is another representation of it. Pizza is a pretty complete food. It has fats, carbohydrates, and proteins. The fats converted to fatty acids, which go directly to acetyl-CoA. The proteins are converted to amino acids, which go directly to acetyl-CoA. Complex carbohydrates are converted to simple sugars like glucose through glycolysis. All of these produce pyruvate, which is converted to acetate, and that goes into the Krebs cycle. So this is just a reiteration of what we've been going over. So this is the emphasis here, and this diagram is the stoichiometry of ATP production. Glycolysis costs 2 ATPs. It generates 4. So there's a net gain of ATP, substrate level ATPs, of 2. Then if you go through all this process, through the Krebs cycle generates another 2 ATPs at the substrate level, but then electron transfer and chemiosmosis generates 32 ATPs. And this is assuming that you start with 1 molecule of glucose. From that, you get a net of 2 ATPs, another 2 here, 32 here, for a total net of 36 ATPs from one molecule of glucose. So it depends on how you count. If you count these 4 as net, you get 38. If you count them, if you count them as gross, you get 38. If you count them as net, you get 36 from one molecule of glucose. So now let's talk about what happens in the absence of oxygen. Anaerobic respiration. And first, we'll talk about what if it happens in a microbe, for example, a yeast. So we start with glucose, and glycolysis gives us, from one glucose, 2 molecules of pyruvate. So from one 6-carbon molecule to 2 3-carbon molecules. And 2 ATP are produced. Remember now, there's no oxygen, so there's no Krebs cycle. And so this is occurring in the cytoplasm. Fermentation occurs in the cytoplasm. So then pyruvate can be converted by yeast to acetaldehyde with the loss of a carbon dioxide. So this carboxyl group here goes off as carbon dioxide, and you go from a 3-carbon compound to a 2-carbon compound, acetaldehyde. Then acetaldehyde can take 2 protons to get this structure, which is ethanol. For example, wine. So this is what happens in yeasts and certain other microorganisms, that you have this loss of CO2 from pyruvate to acetaldehyde, which gets converted to ethanol. Now compare that with what happens in muscle, in our bodies. Again, you start with glucose, glycolysis, get ATP. This is happening in the cytoplasm. But now there is no loss of carbon dioxide. Pyruvate is converted directly to 2 molecules of lactate. 2 pyruvates become 2 molecules of lactate. And that happens by adding protons to this carbon. You go from a carboxyl to a hydroxyl plus another hydrogen. And this is lactic acid. So in muscle, in contrast to microorganisms, in muscle, you don't lose CO2. You convert a 3-carbon compound to another 3-carbon compound, lactic acid. No carbon dioxide is produced. So what is the consequence of that? We've all had the experience of exhausting our muscles and feeling a burning sensation. Why? How does that happen? Well, at the top here, we have this cycle. In muscle, glucose is converted to pyruvate and lactate, plus 2 ATPs. This is anaerobic glycolysis. And this lactate then goes to the liver, where it cycles back to glucose with the consumption of ATP. So what's the sequence? If you're having a high intensity exercise, your muscles run out of oxygen. They become anaerobic. So one of 2 things could happen. Either they can enter into anaerobic glycolysis and have an immediate generation of 2 ATPs per glucose molecule, or they could wait for oxygen, in which case they would get a lot more ATP. But what happens in the moment, in the necessity, is anaerobic glycolysis, because the muscles need ATP immediately. They can't wait for the Krebs cycle. And so the consequence is that anaerobic glycolysis causes lactic acid to accumulate. And this lactic acid in your muscles causes them to feel like they're burning, like they're on fire. So next time you exercise vigorously and your muscles start to ache, you'll know what's going on.
So then theoretically, if you were breathing pure oxygen while you were doing exercise, would you then not create nearly as much lactate?
Probably wouldn't produce any, because if you were breathing oxygen, your muscles, I don't know how fast the transfer would be from your lungs to your muscles, but
Right, yeah, because I'd
certainly,
imagine there's a minimum diffusion time.
yeah, but it would certainly reduce the level of anaerobiosis. You tend to get, if you were breathing pure oxygen, you would certainly get more oxygen into your muscle tissue. But if you're running a marathon, you don't want to take an oxygen tank with you.
Yeah, sorry, teacher. Because of this, during the exercise, the respiration rate is increased to take more oxygen.
Yeah, but if you're using oxygen, excuse me, if you're using oxygen faster than your lungs can provide it, your muscle becomes anaerobic. So that's the whole point here, that if your lungs can't keep up and anaerobiosis sets in, then you get lactic acid generation. And your muscle is rewarded in that it gets some immediate ATP, but it's the cost of that is lactic acid. So that's what this next slide, it shows the two types of anaerobic fermentation side by side in microbes and in muscle. So what yeast does is takes pyruvate and spins off a carbon to make acetaldehyde. And acetaldehyde can be reduced to ethanol. That happens in yeast. In muscle, conversely, this acetaldehyde step is skipped, and the molecule does not spin off carbon dioxide. Pyruvate is reduced directly to lactate. So all this slide is showing is what we've been discussing on the previous slides, except you have a direct, excuse me, a direct comparison here. So the chemistry of cellular respiration, it's very simple at a superficial level. Glucose plus 6 oxygen is converted to 6 carbon dioxides, 6 waters, and 38 or 36 ATPs. Respiration overview. We've gone through the details of this. This simply shows 2 substrate level phosphorylations and then the electron transport chain. This all occurs within the mitochondrion. Glycolysis occurs in the cytoplasm, and pyruvic acid can cross from the cytoplasm into the mitochondrion. And the pyruvic acid goes to acetyl-CoA, and that starts the Krebs cycle. Now, just to state glycolysis, this is respiration. Now, if we just focus in on the glycolysis part of this, it's an exergonic series of reactions, which are catabolic. It occurs in the cytoplasm outside of the mitochondrion. It begins glucose oxidation by breaking glucose into 2 molecules of pyruvic acid, and it makes 2 molecules of ATP, starting with 1 molecule of glucose. And the 2 molecules of ATP come from 2 molecules of pyruvate. So the overall reaction of glycolysis is glucose, 6 carbons, pyruvate, 2 3 carbons. And from this, you get 2 ATPs and 2 NADHs plus 2 protons. You've seen this before. These next 2 slides are the 9 steps of glycolysis, where at first you consume 2 ATPs, and later you get 4 for a net. The stoichiometry, the net is 2, and they're all from substrate level phosphorylation. And finally, just another look at electron transport and chemiosmosis within the inner mitochondrial membrane. We've gone over the details of this. And we are out of time. Any questions or comments about anything having to do with respiration, either aerobic or anaerobic respiration?
So I guess I do. If we could go back to the difference between the 2 different fermentations.
Mm-hmm.
Why is it that in the lactic acid formation, the lower carboxyl group is protonated instead of the electronegative oxygen on the end? Might be a bit too deep for this class, but it was just something that I was wondering.
Are you asking why do mussels do it different from microbes?
I was just wondering about the mechanism for why the pyruvate is turned to lactate versus like having the end electronegative oxygen be protonated instead.
In other words, why does mussel skip the acetaldehyde step?
Kind of. Because my understanding is that the release of the carbon dioxide is a spontaneous process.
I think so. I wondered about this myself. What causes the distinction between what a yeast cell does and what a muscle cell does? And I don't know what determines that difference choice.
I wonder if it's just speed, because you would need to have time for the carbon dioxide to actually be released off, and for the lactic acid fermentation, it just needs to use the pyruvate immediately.
Yeah, could be that it's a rate factor, that muscle is calling for ATP immediately, and so it doesn't have time for acetaldehyde, whereas yeast is in no rush. Yeast is just sitting around in an anaerobic environment. Okay, anything else? Any other slides you'd like to go back to? Okay, then I'm going to stop sharing and remind you that on Wednesday, you'll have all day to take a quiz. The quiz will be on the topic today, respiration, as well as photosynthesis.
Have a good
Okay.
day.
All right. See you Wednesday.